Good afternoon, dear students. Today, Department of Histology and Embryology represented for you the lecture Human Embryology. Greetings in lecture hall number 9. We have seen that's our beautiful lecture hall, and I hope when you start study histology and embryology in lecture hall, you will be satisfied our beautiful auditorium. So first of all, start from the history. The famous English writer Samuel Taylor Coleridge in 1885 was writing the history of man for the 9 months preceding his birth would probably be far more interesting than and contain events of greater moment than all the 70 years that follow it. So during prenatal development, more changes take place than during all humans' life. And we have the beautiful illustration, beautiful pictures of Leonardo da Vinci from his notebook, which can present in the Florence. You have to find maybe in the internet service in some sites. And let's now begin our theory of embryology. The term embryology originates from 2 Greek words, embryon, embryo, and logos, study of science. That's the value of study of embryology to the medical students is 4 aims. Embryology gives an understanding of how the different organs and tissues developed from a single cell zygote into the complex multicellular organism. Second, embryology gives a rational explanation of the relationship and position of many normal adult structures. Embryology includes not only the development of the embryo, but also the development of the fetus membranes which connect the fetus to the mother. Many pathological congenital disorders in prenatal and postnatal development of human possible understood only in comparison between normal and abnormal formation of human embryo or fetus. Well, let's know what is the short story of embryology and basic law and theories. You know, the embryology is a very big science and get it a very long story, but we just investigate during our lecture only some interesting moment that's change the scientific levels and can be discovered the basic development of humans. And we start from theory of preformism. From the time of Marcello Malpighi, Italian scientist and doctor, he was studying that the believed was unfertilized egg in 1675 observed early embryos. As a result, he thought the egg contained a miniature chick. Anton van Leeuwenhoek, using an improvement improved microscope in 1677 and first observed the human sperms. However, they misunderstood the sperm's role in fertilization. They thought the sperm contained a miniature preformed human being, then enlarged than it was deposited in the female genital tract. That scientist, that period, was named animalculist because they too decide that animalculae or spermatozoa, that's the old name, this cell, played a key role in the fertilization. But the opposite, who was think that ovum contained human soul inside, was named ovist. But both of them, unrighted, is absolutely not right for this is talking, because we have development of science, and the greatest scientist who has investigates the process of step-by-step development of own organs from the germ layers, Caspar Friedrich Wolf. In Caspar Friedrich Wolf was been the German scientist, but working in the St. Petersburg University in the time of the Catherine the Great, the famous Russian queen. And Caspar Friedrich Wolf refuted both versions of the preformation theory in 1759, after observing that part of the embryos develop from globules, small spherical bodies. He examined incubated egg, but could not see the embryo described by Malpighi. He proposed that layer concerned the variable division of what he called the zygote, produces layers of cells, now called the embryonic disc, from which the embryo develops. We have seen here his pictures, like this is an embryonic disc with the beginning of the formation of neural plates. We have also seen some areas of chicken embryo and some chicken organs and embryonic organs. But Caspar Friedrich Wolf has ideas that form the basis of theory of epigenesis. He was founded Epigenetic Theory. Epigenesis, which states that development results from growth and differentiation of specialized cells, step by step. This specialization of cells appear like activities of special cells population that time. And now we know this is his name, like differentiation and determination. These important discoveries first appeared in the Wolf doctoral dissertation, Theory Generation. He also observed embryonic masses of tissue that partly contribute to the development of the urinary and genital system. The famous term is Wolffian bodies and Wolffian ducts, now called the mesonephros. It's a secondary kidney, and the primary kidney, and the mesonephric duct, which is named also Wolffian duct, respectively. Well, so the embryology continued developed, and the very famous another person was being the Carl Ernst von Baer. Described the oocyte in the ovarian follicle of a dog in 1827, approximately 150 years after the discovery of a sperm. He has also observed cleaving zygotes in the uterine tube and blastocyst in the uterus. He contributed new knowledge about the origin of tissues and organs from the layers described early by Malpighi and Pander. Von Baer formulated two important embryologic concepts, namely that corresponding stages of embryonic development, that general characteristic precedes the specific ones. It is significant that far-reaching contributions result in his being regarded as the father of modern embryology. And we have seen that the comparison between the old embryo, we have the resembling early stages between different animals to some late stage start the specific phylogenetic changes, especially is very good visible in the late stages, like humans and fetus, rabbit fetus, more change than, for example, reptile and fishes and amphibians. You maybe study this law in the biology. And it was the basic moment for the next famous scientist whose name is Ernst Haeckel, for biogenetic law. Biogenetic law, which is formulated like ontogeny is a recapitulation of phylogeny. That's law Ernst Haeckel formulated with his partner Fritz Müller. But development of this is evolutionary embryological study continued famous Russian scientist Severtsev. Alexander Severtsev, he has to the postulated the principle that prehistorical characteristic palingenesis, like notochord, pharyngeal pouches, cloaca, to replacement by the formation of new organs, like xenogenesis. Like example, new organs or xenogenesis is a chorion, placenta, umbilical cord, and this theory was being completed like theory of phylogene embryogenesis, phylembryogenosis, theory of phylembryogenosis. Well, so the most important for us, this is a theory of critical periods of development. That theory in Soviet Union was being postulated by the Svetlov, Pyotr Svetlov, and the main ideas of this theory was reading like in human development we have seen critical periods, timeline of maximal sensitivity of embryo and fetus for action of external disintegrated factors with the congenital disorders effect. It was been happen around the 20th century. And we have to see this is very important critical periods. The first critical periods of human development, this is fertilization, of course. And when egg fertilize or ovocyte fertilize, it starts a cleavage. And the cleavage is also very important critical periods. Maybe some problems happen, especially we know about monozygotic twins, have to be sometimes result disseparation of doubled cells in early stages because they get the totipotency, like ability to form whole organism. And another moment, this is implantation, when the embryo start implantate, parallel with the process of gastrulation. Implantation is around 7 to 14 days, and this time is possible in other critical periods because sometimes the embryo unimplantate, sometimes this is gastrulation disintegrate, and many young female absolutely not understood that they lost the baby, because this is embryo less than 200 microns. And of course, we have to talk about that period is very important for the continuous development. Next period is named placentation. Placentation, it's if implantation embedding the embryo inside the mucosa of uterus, but the placentation is the formation of placenta. Special child place for joint the embryo and mother organism. Next period is histogenesis, is visible here. We have seen it, very beautiful illustrations how the embryo body is changed. But in that period, ordinary, we have seen neurulation, the formation of neural chip and neural system. And all it's neurogenesis. It's the next period, organogenesis, formation of neuronal system. And of course, systemogenesis continues later, till the periods of 8 months. But the 8 months, it's a normal development, but some early embryo normal birth at 7 months and 9 months late, but at 8 months many fetus was die if was born. Ordinary, we have the birth time after 32 week of development. So let's now talking about classification of all prenatal ontogenesis step by step, and we have seen the beautiful photo of human fetus. We have very good seen the amniotic vesicle, maybe amniotic sac, and also we have seen the chorionic villi. This is rough chorion, also is present smooth chorion, but we will study in the special practical classes of extraembryonic organs, and original fetus, which is now is around the 14 week of development. We have seen his arms, limbs, his head is developed, but not completed phase, as you have seen. Well, so now talking about prenatal ontogenesis. So human development is a continuous process that begins then an oocyte, ovum, from the female is fertilized by a spermatozoon from a male. Cell division, cell migration, programmed cell death, apoptosis, differentiation, growth, and cells rearrangement transform the fertilized oocytes and highly specialized totipotent cells, a zygote, into the multicellular human being. Most changes occur during the embryonic and fetal periods. Look, this is a linear diameter increase from 150 microns till the 50 centimeters in 3,300 times. Weight increase from 1.3 milligram till the 3,000 grams in 2.5 billion times. A cell's quantity is increased from 1 cell till 300 billions in 300 billions time. So what is the early embryogenesis and early prenatal ontogenesis can be divided, and what's the periods, what's the stages. The first stage of prenatal ontogenesis, of course, this is a fertilization. Zygote is the form. This is the first day of development. And the next is a cleavage from first till 7th day. During this cleavage, we have the series of mitotic divisions, but without growth. and the diameter of cells is decrease, decrease, and decrease. Result, we have a blastocyst. That's diameter of a whole blastocyst is the same as zygote, around 130-200 microns. Gastrulation is a next period. This is a unical stages of transformation of embryo body into the future fetus. It's happened in 8 to 23rd days. During this process, we have formation of axial organs and gem layers. We have also seen here, this is a zygote, this is a blastocyst, this is a layers and axial organs. Primary histogenesis, it's around the 24th to 56 days. That's formation of tissue rudiments. And we have the next period is definitive histogenesis from 9 to 38 weeks. System of organ is formed, and the 38 weeks is present neonatal newborn stage. Neonatal stage, that's newborn babies, ordinary investigates and treatment by special physician, which is named neonatologist. Look at these full stages. We have seen here a very important moment, that each of these stages characterize a special period of development. Look, this is the first day fertilization and second period, that's cleavage, first to 7th day. Zygote and blastocyst is a common name is conceptus. Conceptus is not fetus, is not embryo, it's not maturated embryonic period, because early embryonic period, which is named conceptus, zygote and blastocyst. But when is it happen implantation and gastrulation start, we is named this is period from gastrulation till primary histogenesis, 24th, 56th day, until the 8th week of development is named embryonal period. Embryonal period. Till 8th week of development commonly. And after that, we have talking about definitive histogenesis, when from 9 to 38 weeks is start the formation of system of organs. That period is named fetal period, fetal. Of course, if the zygote and blastocyst is named conceptus, that's the embryo. This is the period from 8 day till 56 day, embryo. After, from 9 week, we're talking about the fetus. And this is fetus continued period of fetal development, continued till the neonatal stage, around 38 weeks in normal neonatal stage. Well, what is it? Primordial gem cells. Primordial gem cells. This is a rudiment. This is a precursor of gametes. Common features. That's a haploid, chromosomes number. Early originate from endoderm of a yolk sac, And we have seen here this is endoderm yolk sac wall. Except endoderm and the yolk sac, we have also mesenchyme, and mesenchyme is not participate in this process, but endoderm to give rise to formation of primordial gem cells. This is gem cells is migrated into the gonad region, special organs, where is to transformated into the future ovagonia or spermatogonia. So this is specialized generation of cells which is characterized meiotic division in a region. Special population of cells, generative cells. You need to write this information necessary. I want to tell you, my dear students, that during our lectures and practical classes, I will inspect it. How do you write your lecture? And I will be waiting how do you will send me the photos of your lecture, maybe show your lecture notes in practical classes. So, next moment. First type of sex cells is a sperm cells. Spermatozoa is a male cells or gametes. And this is, we have seen here the two mechanisms. That's one mechanism, it's scanning electron micrography. We have seen here the head of sperm cells, but the tail is not be visible. And another, this is a classical histological stain, iron hematoxylin. And we have seen very good visible acrosome and head with nuclei, neck, and of course principal piece and end piece. So this is very interesting. That flagella is very good visible in classical optical light microscopy and not visible under the electron microscopy technique. Look, the structure of the spermatozoa. Ordinary is very good visible. That's diameter of spermatozoa 70 micrometers. That's velocity, that is movement of spermatozoa around 30 to 50 micrometers per second. It's one of the more faster cells in nature. And we have the special structure which is represented in this picture. Sorry, that's in Russian, but we have seen here the receptors of sperm cells in the tip of the head, which is also visible acrosome number 2, and also we have seen the nucleus inside. Also we have seen the neck. Also we have seen the middle piece, middle portion with mitochondrion, and main portion and tail. But more correctly, special structure is visible here, is very good seen the fluorescent microscopy, or another variant is named confocal microscopy of the head of sperm cells. And again, watching the English prescription. So first of all, this is a nucleus number, here is visible acrosome, and centrioles. Centrioles, we have double centrioles. One centriole is named proximal, and another is distal centriole. Proximal centriole to using for migration, in future migration into the oocyte during fertilization. That's very important for completed the second meiotic division of oocyte. Why we have discussed later. And another centriole to make the base for axoneme, the central tubules of flagella. And we have to see also another component of spermatozoa body. After the centrioles, we have the mitochondrial shift, is very good visible here, like spiralized at the lateral surface. Also mitochondria and axonemal complex in the center. Fibrous sheath, it's present like surrounding elements. This is axonemal complex, and this is fibrous sheath. And we have the membrane, surrounding membrane. And we very good seen the examination electron micrography. This is micrograph to show the bat spermatozoa. This is not humans. This is a spermatozoa of bat. But we very good seen the elongates nuclei. We also have seen the cross-section of flagella, very resemble on cilia, because again formula double on 9 plus 2. And this is a classical microtubule aggregates which is joined with Dennean arms. Well, thank you for talking about spermatozoa. Now we're talking about the progenesis of spermatogenesis. What is it? Progenesis. Progenesis, this is a word, terms which we use for gametogenesis in embryology. We have divided the progenesis into the spermatogenesis, formation of male sperm cells, and oogenesis, or ovogenesis, the formation of ovocytes. And what about consideration of male events? Look, we have first of all started from the testis. That's a testis, it's a germinative organ of male which can be seen like a system of original testes, like glands, male gland, and specialized edit part which is named epididymis. Epididymis to provide the maturation and provides the activation of the sperm cells for future fertilization. And we have to see that during this process of maturation, the inside epididymis, the surface of the sperm cells, is a covering by special proteins we have to discuss later, and flagella will be active because before migration to epididymis is not active. For that, we need to around any days for activation of fertilizing ability. Like first of all, spermatogenesis, as you know from biology, can be divided on some phases. The first phase is multiplication. Multiplication, this is process of mitotic division. As a result of this multiplication, we have talking about the formation of spermatogonia. One of these is more dark, another is more light, but we name this spermatogonia A and spermatogonia B. Next stage is named growth phase. Growth phase, ordinary, this is meiosis. It continues around 22 days till the maturation phase. And thus very important to know that spermatocytes type 1 as well as spermatocytes type 2 is result of first and second meiotic division. And effectively have seen the spermatids like not full maturate cells because it's not too dissipated the extra cytoplasm from the flagella. But during formation phase, the phase number 4, ordinary is absent in female oogenesis. During this formation phase, we have the formation of flagella and formation of acrosome and acrosome enzymes from Golgi complex, from Golgi apparatus. So acrosome, this is modified Golgi complex. As effect of that, we have number 5, this is a classical spermatozoa. Again, multiplication phase, this is mitotic division. That's the next is a growth phase, meiotic division. Next, this is maturation and formation. As a fact, we have seen the normal spermatozoa. That stage is also very good, visible here, especially this is the time of maturation and formation, when we have seen how the spermatids to transform to spermatozoa. Continue. The spermatozoa ordinary can be identified in the spermogram in the analysis of a seminal liquor after ejaculation. Ordinary, we have 3.5 milliliters of ejaculate, but we have seen that's around 20 to 200 million per milliliter sperm cells can be identified inside this is seminal liquid, seminal fluid. And that's a normal spermatozoa is ordinary 60%. Unmature around 2%. Leaf cells 75%. Mobile cells more than 50%. If this is parameters correct, we're talking about normal spermogram. If this is parameters less, like example, mobile cells less than 50, maybe only 20%, it will be problem, it will be disease pathology. If normal cells less than 60%, it's also pathology. Example, first of all, aspermia without cells absolutely. Next is oligospermia, if the cells not enough, less than 20 million per milliliter. Next, this is teratozoospermia, if the cells get it 2 head or 2 tail. This is pathology. Also, teratozoospermia is no abnormal structure of sperm cells. Next, this next is anisozoospermia. Anisozoospermia if a sperm cells is not moved absolutely or move very slowly. Like example, these short sperm cells or maybe these very tiny not active sperm cells can be fertilized egg. Why that parameters can be changed? Why the normal cells and mobile cells decrease? First of all, this is time poison, like example, alcohol intoxication, nicotine intoxication, narcotics can be identified after 30 minutes after smoking or drinking in the gonads. And another moment, of course, this is needable special time for normal spermatogenesis. It's an ordinary 72 days. But in different seminal ducts can be developed the sperm cells in different time. It's asynchronic. Convoluted ducts can be produced the sperm cells. And we have for decision that's problem of infertility, male infertility, and sometimes female infertility also happen. We have a special equipment. That's a special equipment is named devices for in vitro fertilization, is represented in this picture, in this photo. Like a famous mechanism of in vitro fertilization was being provided by the English scientist Edwards and Steptoe. That method is named Intracytoplasmic sperm injection. As you have seen here, this is oversight, this is of course artist work, but we have seen how the micromanipulator penetrate the ovalemma and injected inside ovocyte cytoplasm sperm cells. That method is more famous after the work of Edwards. And we have that person, that's a scientist, Edwards and Steptoe, get it the Nobel Prize in 2010. And also another method is named hatching, supporting hatching. When embryo after fertilization, we got the stage 8 blastomeres, and we have removed one blastomere from the provide normal implantation. So removement of one blastomere not disintegrate embryo, but can be activate the process of implantation. Ordinary, that's did it with 4 or maybe 5 8-stage cell embryo. Well, thank you. Continue the oogenesis, the formation of female sex cell, and again same stage like phases: multiplication, mitosis, and the second growth phase is meiosis. We have specificity like small growth ordinarily take place in embryogenesis, large growth in puberty, a maturation phase meiosis, and a prophase 1 for female oocyte. We have stationary stage in diakinesis. It means that stop the development anytime ordinarily happen in small growth. And orgonia, 2 oocytes 1, oocytes 2 type, it's happened during the problem with centrioles after the formation of polar bodies. That's a metaphase 2, is a stop phase without centrioles. Look at this picture is shown for us. That's an important moment. First of all, we have seen that during oogenesis is visible polar bodies formation. Why we need the polar bodies during female oogenesis? Because that polar bodies is very needable for regulation of future cleavage. The 3 polar bodies to operate the vectors of cleavage after fertilization, and they located in the spaces between the fertilizing envelope and ovalema. Also, we have seen the maturation only one cells from 4. Only one cells from 4 is maturated to oocyte, secondary type, but in other it's a polar bodies. One moment we have seen here the comparison between the sperm cells formation, spermatogenesis, and of course oogenesis. That's happened inside specialized organ of female body, which is named ovary, and in ovary the morphofunctional unit is follicle. As very good seen, primary oocyte and primary follicle, primary oocyte in a multi-layer follicle, secondary follicle, and we have a Graafian follicle with antral spaces. Maybe you study in the school, maybe you study in biology, but the main idea is that this is organs, this is a specialized unit of ovarium, or another name is, follicles, play the key role for the maturation and formation, division, meiotic division of the oocyte. In this picture, in this photo of microscopic photo, we have seen Graafian follicles, and we have seen the endoscopic photo of multi Graafian follicle. That's ovary with numerous tertial follicles. Tertial follicles is Graafian, which is mature follicles, and they are represented here before the ovulation. We're not talking about long time, but look at this artist work. We have seen here all stages of development of follicles from the primary till the secondary and Graafian, and after that we have seen the ovulated follicles and corpus luteum. Corpus luteum played a key role for the production of hormones, especially progesterone, under the controlling from the hypophysis hypothalamus by luteinizing hormone. That's pituitary gland synthesize luteinizing hormone, and ovulation and future formation of corpus luteum take place. Well, so in this table with some diagrams, we have seen the early development of ovarian follicles, menstrual phase, proliferative phase, some completion of development of follicles. We not spend the time for that because, sorry, we have a very long lecture and many other embryological questions. But I want to tell you about this moment. That's all this is process of maturation of follicles and ovulations, migrations of ovocytes in uterine tube play the key role for the future fertilization, because it's very important for the day. What's a day after fertilization? The zygote migrated and maybe, blastocyst, this is late blastocyst, migrated into the wall of the mucosa of endometrium, of uteric mucosa. Because if it happened after 24th day, maybe not enough time for have to fully complete the process of placentation. Implantation maybe, but placentation is not developed. As a effect of that, that is not happen process of pregnancy. Another problem when before 80 days will be not possible the normal fertilization because it's maybe not can maturate the surface epithelia in uteric wall. Like example, for the normal implantation, we need a specialized cells, decidual cells. It's modified fibroblast in uteric wall, in endometrium. And if this is decidual cells is not formed, also implantation is impossible. But more important, we will study these questions in the topic of female reproductive system. Let's now back for female gametes, oocytes, and we have seen electron scanning microgram, which is coloring in Photoshop. It's very good visible that this is oocytes located in the special spacula, maybe very small, maybe like a spatule, yes, and we have also visible the specialized cells, follicular cells, supporting cells of ovum, and we have seen the beautiful, like incrustation, Zona Pelutzida. This is a Zona Pelutzida 3. Type 3, this is a outmost surface, which is very rich by the some glycoproteins and has a very unique structure, maybe like in jelly handmade. But that's not handmade, this is a nature made. And we have seen the follicular cells in ordinary fluorescent microscopy. And also back for the stages, like human oocytes. How we classified human oocytes? Human oocytes is a secondary oocytes. Oligolecital, secondary oligolecital, and isolecital in shape. So in relationship between yolk granules and spaces inside. So we have seen the fertilization site of uteric tube. This is ampulla, and that's a site which located near the outermost tip of the uteric tube near the ovarium, which is ordinarily has special elements, fibria, and this is fibria ordinarily like touch the surface of Graafian follicles before the ovulation, to activate the area of ovulation, which is named stigma. And why we name the ovocytes after the ovulation, after the entering from the mature follicles? Why we name secondary oligolecytal? Oligolecytal is mean less yolk granules than another type. Like example, chicken egg is named polylecytal. But humans lost the quantities of yolk granules and has another organization because get the development inside the uterus with a specialized organ in future placenta, which provide the trophical effect for future embryo and fetus. Well, next now talking about the structure of oocytes. Human oocytes receptors. We have seen, this is schematic diagram of picture of oocyte, and very good visible the cells which surround the human's oocyte. This is cells is named follicular cells, but the envelope which constitutes from the follicular cells is named corona radiata. That's beautiful name. Corona radiata was named Carl von Baer. Von Baer to have seen this oocyte under the light microscopy, and his very beautifully illuminate because the light migrate through these cells. And Baer to talk this is like a sun because it's named corona radiata. As another moment, this is next envelope which is named Zona Pelutzida or brightness zone. Zona Pelutzida. Pelutzida is mean like bright. That's also like illuminations. But Zona Pelutzida consists from three layers. The first layer, this is ZP2 layers, is located near the ovarium. We have seen ovarium like projections of some microvilli which is joined with follicular cells, interactions between ovarium and follicular cells. ZP1 is located between ZP2 and ZP3, and the outermost is named ZP3. ZP3 layer, that's receptors of human oocyte. That's a special name of this special substances of human receptor is named N-acetylglucosamine. Only human oocyte characterize this is biochemical substances. Like example, cats or maybe amphibians oocytes get in absolutely another substance, not acetylglucosamine. And we have all structure of these envelopes in our slide. Next, this is a classical vision of oocyte under the microscope, light microscope, and you will see it under the microscope in our practical classes. As you have visible also follicular cells here is a red color, and the Zona Pelutzida it's a blue color, nucleus it's a purple color because that's a special stain. It's Mason trichromasia, tricolor by Mason, and Mason, sorry. And this is trichromasia to have been identified different layers of the oocyte. Well, next, what is fertilization? So let's be ready for writing now. You know that fertilization is the act of fusion of male and female gametes to form the zygote. The fertilization takes place within the body of female and normally occurs in ampulla or ampulla-isthmus junctions of uterine tube. I just tell normally because sometimes it's happened abnormally. In this case, the fertilization takes place not in the ampullaric region, but maybe in another part of the uterine tube, sometimes outside the uterus, and maybe is present in the spaces between uterus and rectum, like as famous is Douglas space. So continue our moments. This is fertilization can be classified in some special part, like distant interaction, capacitation. That's a distant interaction. This is the first step of fertilization. Capacitation is continued around 7 hours. Capacitation involves the release of epididymal fluid, glucoconjugate, from surface of head of sperm cells. This is glucoconjugate special proteins, glycoproteins, which can be removed as effect of rheotaxis. When sperm cells can be migrate against the flow of female female secretes, like its ordinary, it's mucus. Sorry, this is a special mucus which is produced by the neck glands, in cervical glands in uterus, and of course, and also some glands, some cells in the uterine tube. Effect of it, we have the process of rheotaxis. But the sperm cells also migrated to the area by chemotaxis. Chemotaxis as effect of chemical stimulation stimulates the fertilization. Like example, we have the henogamons. Henogamons is hema-attractant, which actively attract the another special cells, like sperm cells, into the site of fertilization. Example, we have two uterine tube, left and right, in which uterine tube oocytes waiting the sperm cells, in left or in right. It's possible only with hemotaxis, with a activation of like special signals. And this is special signals, this is henogamons. One moment, sperm cells also synthesize androgamons, and oocytes is known as any sperm cells migrated in this area or not. Continue our talking about the fertilization, how many cells take place in this process. That we have the 200 sperm cells which migrate into the area of ampullaric region. You can ask, what about another billions of cells? But another cells can be dying on the pathway because the mucous surface of the female reproductive tract ordinary is lubricated with a special lactic acid, and this is lactic acids to can be kill the some cells. But of course we very important that we need that around 200 cells migrated inside the ampullaric region. So total quantity of the sperm cells is 300 millions, but only 2 millions in ampulla migrated and take active part in fertilization, 200. We have 96 sperm cells has X chromosome, and 104 sperm cells has Y chromosome. Because ordinary the boys is often to create, but many of them is dying. Some embryo is dying, some is dying in the time of the fetal time, and sometimes that's a newborn babies in neonatal time. Also some boys is dying because that's a problem of critical periods also. Well, in the next moment, what about stages of the fertilization? The first, as we're talking about, that's capacitation, 7-hour continued, contact interactions and rotations continued around 12 hours. This is rotation take place when sperm cells aggregate surround the ovocytes and start to rotate, move the ovocyte. Next is acrosome reaction. Acrosome reaction, that's a process when sperm cells is release the special enzymes from acrosome. Ordinarily, this is hyaluronidase. It's enzymatic factor which destroy the Zona Pelutzida. All three layers, ZP1, ZP3, ZP2, all is destroyed by hyaluronidase. As effect of it, the sperm cells can be migrated inside into the ovolemma, and that's process of reduce the follicular cells, open the ovolemma, is a name like process of denudation. So again, stages. First is capacitation, rheotaxis. Next is contact interaction, rotation, acrosome reaction, denudation. Next, this is again, we have seen fertilization is visible here. Capacitation, acrosome reaction, zona reaction, and cortical reaction. Zona reaction, this is a process when sperm cells migrate inside the ovocytes. But the cortical reaction, when the effect of calcium ion wave, the cortical granule is a start exocytotically release special substances in perivitellin space. Perivitellin space, this is a space between the ovolemma and Zona Pelutzida 2. Effect of it, we have the formation of fertilizing egg, fertilizing envelope. So the very important moment, this is impulse activation. Impulse activation is appear like a cortical reaction effect. Look at this, we have seen Zona Pelutzida, migration of sperm cells, and when it starts the wave of calcium ions, as it starts the activation of releasing of cortical granules, perivitellin space is visible, the formation of blockage, the polyspermia, like effect of zona reaction and cortical reaction. As the effect also visible here, like combinations of ions, dielectrical potential, which to change the condition of fertilizing egg. After that, with very good visible here, how is possible, how it is continued to develop. And we continue study the main moments of the summary, like summary of fertilization. During fertilization, the spermatozoon must penetrate the corona radiata, the zona pelucida, and the oocyte cells membrane. As soon as the spermatocyte has entered the oocyte finishes its second meiotic division and forms the female pronucleus. The zona pelucida becomes impenetrable to other spermatozoa, and the head of the sperm separates from the tail, swells, and forms the male pronucleus. The results of fertilization are restoration of the diploid number of chromosomes, determination of chromosomal sex, and as initiation of cleavage. So talking about this is moment, we have seen this is fertilizing envelope, we have seen the oocyte cytoplasm, and we have seen two pronuclei, large, this is female pronuclei, and small is male pronuclei. That process is named conjugation of pronuclei, also named is process of formation of future zygote. And we have seen the area where is sperm cells immigrated inside with the micropelase name, and that process is very good visible here, whereas we have seen the envelope, fertilizing envelope, and polar bodies under the envelope. Again, repeat all stages: distant interaction, capacitation, in contact interactions, rotation, acrosome reaction, denudation, penetration, cortical reaction, zona reaction, and cleavage, preparing for cleavage. Well, so continues our talking, and we have to a little late just the definition of cleavage. Cleavage is a series of mitotic division that results in an increase in cells blastomeres, which become smaller than each division. After 3 divisions, blastomeres undergo comparison to become a tightly grouped ball of cells with the inner and outer layers compacted. The blastomeres divide to form 16 cells morula. As the morula enters the uterus on the 3rd or 4th day after fertilization, a cavity begins to appear and the blastocyst forms. The inner cell's mass, which is formed at the time of compaction and will develop into the embryo proper, is one of pole of the blastocyst. The other cell's mass, which is surround the inner cell and blastocyst cavity, will form the trophoblast. Look at the next moment. Look, this is a cleavage. We have the 2 blastomeres, 4 blastomeres, 3 blastomeres, 4 blastomeres, 8 blastomeres, 16 blastomeres, and all these stages is very good visible here. Well, next, so humans' cleavage can be characterized like full unical asynchronic, two blastomeres first to form the black and white blastomeres in time of the days and more faster 12 cells. 50-60 hours we have talking about the 16 cells morula, and the 5th days is named blastocyst. And we talking about the cleavage of first division, 300 hours, 30 hours, 2-cell stage, second stage is 40 hours, 3-cell stage, 3-4 division, 4 days morula, and 6-9 divisions, 3 blastocyst, and next this is the implantation will be 7-8 days, which is named as Tetrablastula. After that, conceptus transformed to the embryo. In this schematic picture, we have seen the all stages of migration and fertilization and migration, zygote, 2-cell stage, 4-cell stage, 8-cell stage, morula. By the way, 8-cell stage is more important for in vitro fertilization. Ordinarily, that stage was made in the petri dish, like that, maybe like phraseological phrase is, maybe have to use this stage for implantation artificially. But what about the blastocyst? Blastocyst ordinarily take place during the normal development, and it contains more than 32 cell mass. This is 8th cell stage, very popular for experiment, and structure of blastocyst, formation of blastocyst, cleavage and blastocyst. In blastula of mammalians, animals and humans, it constructed from blastomeres. Blastomeres, this is separated cells, and blastoderm, which forms the inner cell mass, dark blastomeres, embryoblast, which is surrounding by the light blastomeres, trophoblast. Look at this one. This is a embryoblast, this is a trophoblast. Embryoblast, trophoblast. And we have seen here the electron scanning micrograph of blastocyst with a modification with Photoshop coloring. Trophoblast, first extraembryonic organs. Outer cell mass became flattened and are collectively called trophoblasts because of their play embryonic nutrition. Trophoblast temporary organ. Layer represents in the 5-day precursor of fetal component of placenta. The conceptus at this stage of development is called blastocyst. The wall of blastocyst is called blastoderm and is composed of trophoblast. Trophoblast consists from 2 layers, outermost. This is syncytiotropoblast or symplast trophoblast, that is result of multinucleated non-cellular structure. And inner cell mass is cytotrophoblast or Langhans layer. This is single cells, flattened cells layer. And all this is cytotrophoblast cells produce chorionic gonadotrophin in time of 5 days of development after fertilization. This is chorionic gonadotrophin represented in the wall of the uterus. This is moment of implantation. This is a blastocyst which is implanted into the mucosa of endometrium. And we have the beautiful visible structure of this area under the electron scanning micrography. During the formation of chorion, primary chorionic villi, this is the 70-day embryo. Crimea chorionic villi is visible like system of syncytiotropoblast or symplast trophoblast. and cytotrophoblast, without blood vessels but with visible specialized tissue mesenchyme, which ordinarily arise from extraembryonic mesoderm during development. So this is villi to provide normal feeding of a future embryo via the formation of future placenta. But this is only one of part of it. This is electron microscopic photo, which is shown for us syn situ or synplast trophoblast, multinucleated part, and cytotrophoblast with mitochondria, round nuclei, and some basal lamina. Also can visible vessels inside. Well, but we also have seen wonderful, the earliest embryo in Earth. That's result of our investigations in Crimean Medical Institution. The famous Professor Yuri Shapovalov and his students and assistant Barsukov can be made the investigations of this embryo in the beginning of 1970. We have seen here this is placenta formation with a villi. Also is very good visible notochord. Sorry, it's not is a primitive knot. That's a period of the formation of gastrula, first gastrula. And also we have seen amniotic fluid. This is primitive yolk sac formation. And well, you need to know that this is embryo, the early stages of human embryo in history was made in Crimean Medical Institute. So what is it? Implantation. Implantation, Latin implantation is in-growth process in 6-7 days in upper segment of uterus. Identified 2 stages, adhesion and invasion. After the invasion, the primary chorionic villi is formed in the 14 days. A chorionic gonadotrophin is identified in urine around the 14 days. So you need to remember adhesion, first stages, like sticking for the wall of uterus. Invasions, it's like embedding into the wall of uterus. And under the control, this chorionic gonadotrophin, which activates the transformation of endocrine system of female for pregnancy. And the chorionic gonadotrophin influenced into the hypophysis and hypothalamus for change the activities of luteinizing hormone for support the corpus luteum in the ovarium to transform to corpus luteum of pregnancy during the 9 months of development. Well, and so next, this is process, how does it happen? So we have seen again trophoblast layer, inner cells mass, embryon blast, and we have seen the formation of chorionic villi, primary chorionic villi, maybe like projection of trophoblast, synplast or syn situ trophoblast into the uterine stroma and epithelium. Well, during the implantation start the gastrulation. We have seen the first stage of gastrulation, which is named delamination, the second stage, which is named immigration. So it's possible only as a result of very important process of determination. Development proceeded in a series of stages in which precursor cells are first restricted in their developmental potential, determination, and subsequently express their genetic information as specific tissues, differentiation. During early embryological development, cells are said to be totipotent because they have the ability to produce any and all of the cells that are found in the adult organism. However, as the cells continue to divide and develop, their developmental potential begins to decrease, and eventually their fate becomes fully determined in process called determination. So this is a restriction of ability to cells for development all direction except one. And cells determination is the process by which the cells commits to the certain developmental pathway and eventually produces a specialized cells. Cells determination usually takes place as a result of inductive signaling between nerve cells. Look at this one. Determination is genetical restriction, differentiation, this is specialization, this is a expression of special genes, and it's possible to regulate through interactions, intercellular interactions, via signaling molecules and growth factors. So, differentiation is shortly we have, say, specialization of cells. There are the three different mechanisms by which inductive signaling can take place. Following the cell's determination, this unspecialized by determinate cells undergoes a series of steps and processes that produce a specialized cells, and these cell steps are collectively called the cell's differentiation. Differential gene expression allows the two identical unspecialized cells to follow two different pathways and produce two different cells. Example, next, the realization of this is differentiation, and determination is gastrulation. Gastrulation is the process of formation, the three embryonic gem layers, ectoderm, endoderm, and mesoderm. You need to remember this very important definition. Again, process of formation of three embryonic gem layers, ectoderm, endoderm, and mesoderm. So we have seen that the embryo is located inside the uterus through the special envelopes, like one of them is the decidua envelopes, and another, this is placenta and chorions. We have the formation of placenta barrier in the future, but we not have time for talking about it now. With embryonic gem layers and external organs, which you need to know. First is ectoderm, outmost skin, outer gem layer. Endoderm, inner skin, inner gem layer. Mesoderm, middle skin, middle gem layer. Mesenchyme was appear in 11-12 days, provisory embryonal rudiment of connective tissue. And also we have the axial organs like neural tube, notochord, and primitive gut. As during the differentiation of the embryo, we have the special differentiation of each of these gem layers. Like epiblast gives us the formation of ectoderm of amnion, embryonic ectoderm, and primitive strake ectoderm. Also, that's a primitive strake is not ectoderm, but it's initiate from ectoderm. We have the formation from primitive strake extraembryonic mesoderm, embryonic mesoderm, notochord process, and embryonic endoderm. But embryonic endoderm, it's arise from as a result of gastrulation. That's a hypoblast, gives us the formation of endoderm of umbilical vesicle and umbilical cord, and extraembryonic mesoderm. So, what is it? Formation of axial organs. We have seen here neural tube, this is notochord, and that's somite. Somite is a differentiated part, paraxial, lateral mesoderm. We have also seen the implantation, during the implantation take place first stage of gastrulation, delamination, the separation of the embryonal disc into hypoblast and epiblast in 8 and 7 days. Gastrulation in 9 to 12 days is named immigration, and as effect of that we have formation of embryonic mesoderm and in embryonic organs, and also we have the 12-day embryo. This is delamination. We have seen that's formation of hexacoelomic, hexacoelom, and it's provide the formation of chorionic cavity, and extension of amniotic cavity. And yolk sac is start to transform to secondary yolk sac. This is a process of migration of cells during gastrulation, second phase, immigration. As we have seen here is one plast of cells is migrated through another. This area is named primitive knot or Hansen knot. Next, this is gastrulation immigration, 15 days. We have this primitive strake, is a forms like migration of cells from epiblast and hypoblast and to form the middle layer. Mesoderm is very good, visible here. Entoderm, innermost, and ectoderm is more bigger in thicker in the outermost. So this is future epithelia, this is a future muscle and bone, and this is a future gut and intestine. So we have seen it here: endoderm, ectoderm, mesoderm, endoderm immigration, and we have the prechordal plate formation of future ectoderm and endoderm, and we have the neurulation, the 70th day of neural plate, the separation from the surface ectoderm, and neural tube under the inductions which have been seen. Somatogenesis, the formation of somites to form the special inductional pressing from the surfaces of lateral surface and to close the neural tube. Caudal and proximal neuropore is present, and we have the neural crest formation in 19th day. This is neural crest. It's a group of cells which is migrated in different areas. And we have to discuss in the practical classes what organs is arise from neural crest. And summary: 17-day neural plate, 18-day neural groove, neural tube, 19-day neural crest, 27-day spinal cord and brain formation. We have seen again somites like mesodermal condensation, and we also have seen this is a paraxial mesoderm, intermediate mesoderm, and lateral splanchna-pleuric mesoderm. This is a component of mesoderm. Is very good, visible also here. And last, this is derivatives of each of these areas: ectoderm, mesoderm, and entoderm. You will need to study in practical classes. So during continuous development, the body of embryo is very fast to change. We have seen cranial neuropore, we have seen a primary heart, and we have the embryo around the early 10 days of development. So we have the cross-section of embryo, and thank you very much. Our lecture is finished. I hope you can continue the study of embryology in the practical classes. If you need to ask me, please, I will waiting your asking question in the chats of my groups. Okay, thank you very much.